Hello and welcome. Welcome to those of you who are visiting NITMB. So let's see. My name is Elizabeth Jerison. I'm a member of the physics department at the University of Chicago. And I was asked to give a tutorial about things related to inferring dynamics, inferring dynamical systems, so I should say that I am actually relatively new to this topic. My group studies immunity, and we are primarily interested in inferring dynamics in that context. But because the lion's share of the work on this topic has been done on essentially single cell RNA sequencing data, that's primarily what I'll be talking about for sort of pedagogical purposes today. So the subject here is, as it says, is inferring dynamics from time-resolved cross-sectional data, and you should really jump in with questions, because it will be more interesting that way. So what I'm going to do is first talk about what types of problems, or at least give at least one specific problem that people are trying to solve with these methods. Talk about sort of what I would say is one of the primary classes of methods that are used, which are types of diffusion models, and why, and just give a survey of there's been quite a lot of recent methods development in this area, and then I'm going to go into just a little bit more detail about how one of my favorites of these works, and this is a recent reference and really a nice paper, and then talk about some questions that perhaps might be interesting to you about the relationship between this and sort of other slightly buzzy topics that you might have heard about, and then end with just a brief mention of other things that might be worth reading about along these lines. So what problem are we trying to solve? So as I'll mention later, in some sense, some of these modeling approaches I think apply to a number of scenarios beyond single cell RNA sequencing or beyond cellular state transitions, but partly because that's the focus of this workshop, and also, as mentioned, because it's really where many of these methods have been developed in this bio space, at least. I'd like to sort of ground this in an example from cellular differentiation. So the problem is that you have, for example, a hematopoietic stem cell, HSC, which can become many other types of cells, right? So this guy here, importantly, this guy here, this progenitor here, is not just a single sort of monolithic identical cell. There's small differences in different numbers of proteins, mRNAs, and other sort of chemical constituents of this cell, right? So this cell, this progenitor, still probably has the ability to become, potentially other cell types, but there are, based on its initial state, there are biases, and at the end of the day, so it will undergo a sort of largely autonomous process in which it can differentiate into, in this case of blood cell differentiation, you can get your different populations of monocytes, neutrophils, and basophils, for example. So that, for example, if you have some kind of a characterization of your cell at the initial time point, then maybe at the end of the day, there's some probability for its descendants to become each of these 5 different fates pictured here. So there we go. Right. So underneath the hood, what is happening to determine how the cell differentiates? So the picture. So a simplified picture of what's happening is that there are a number of regulators of gene expression, which then drives, differences in proteins and so forth, and eventually the function of the cell. So many of these things are regulated by sort of networks of transcription factors. And the idea is that this drives autonomous dynamics within the cell. So, you have some levels of all these regulators to start with, and then, they will promote the growth or decay of each other in some perhaps quite complicated way, as pictured in this little wiring diagram here. But the suggestion from this is that maybe something about this initial state determines the subsequent time evolution of the system. But as mentioned, this is already a relatively high-dimensional and complicated dynamical system. It's basically hopeless to parameterize all of the actual functional connections along these arrows in any cell type, every cell type you might be interested in. And additionally, there are various sources of noise. So in reality, one of the, people often think about things like transcriptional noise and, numbers of mRNA molecules, et cetera. In reality, I think, when people have gone and measured this, the majority of the noise or the vast majority of the noise is really not that. It's really essentially has to do with hidden biological variables that we're sort of not measuring, but that are sort of effectively, can be effectively considered essentially sort of random variables in any given cell. But it's clear that, it is not the case that, if you measure, again, what in these contexts, people are going to be typically measuring RNA. If you measure, the levels of all the RNAs, that doesn't, it really only probabilistically tells you what will happen to the cell in the future. Questions? But the question is, okay, so given that you have this, you have underneath the hood somewhere, you think you have a complicated dynamical system that maybe can be modeled as something where we have some deterministic parts that we know, and then there's some stochastic components. But we really don't know, what are all the right equations to write down. So can we learn anything about these landscapes? Can we learn anything about these landscapes directly from data? So this is just a sort of an artist's rendition of sort of an actual realization of what is often called the Waddington landscape from a recent paper, also a nice one that does one of these things. This is again blood cell, so HSC development into monocytes and neutrophils in this example. And so the question is, can we learn something about this underlying landscape? And so in some of these methods, as I'll get to in a moment, in some of these methods, there is an actual what is essentially a potential landscape that is actually inferred. Some of them do not require that the sort of underlying structure is summarizable in that way. I'll mention that in a second. But in some cases, there is essentially inference of a potential energy landscape going on in the background. All right, so what are we trying to do here? So the idea is that you'd like to be able to make predictions about, starting from some point, what kinds of cells will you get. You'd like to understand something about the effects of perturbations. Maybe you want to make more monocytes or something. You want to actually be able to engineer and steer the system. Common types of perturbations that people try to introduce, some of them are essentially sort of gene knockouts, because it's something that can be compared to actually done experimentally and compared. Ultimately, people also have a lot of interest in, for example, if you put in some kind of regulating factor, what can you drive cells to do? And then finally, and interestingly, people would like to be able to use features of such an inferred landscape to say something about the underlying biology and what, for example, what transcription factors and interactions are actually driving key transitions. And what I'm going to talk about, basically limit myself to these examples where you have these types of destructive measurements. So you sample your cells at a particular time point, or your cells or your whatever it is, but you do in fact have an actual time series. And this is for these types of methods, as I'll allude to, and I think and will come up more in the next talk also. This is important. Okay, so to motivate the sort of main modeling approach for these instances, I want to go back to this concrete example of a gene regulatory network to start with. Why would you use something like a diffusion model? So if you for example, try to write down what might happen, how the levels of these different genes here might influence one another, you could start with a stochastic model, something like this, where there is some rate at which species X gets another molecule, and this is determined by, again, all the levels of all of the different regulators, which in reality are probably proteins, but the hope is that the mRNA levels of those different molecules are a reasonably good proxy. And then in this formulation, there's also V, which is the reaction volume in the cell. So G here is an as yet unspecified, so just any some kind of nonlinear function that takes in all of these inputs of this the state of the cell and tells you at what rate then that means that GATA2 is reproducing. And then there's also just some decay. So if you write down the continuum limit of this model, and in this case by continuum I mean that we can treat now X, the number of molecules of a particular species, as a continuous variable. So this, you can think of this simply as the concentration. And so this is, valid as long as there's, sort of a medium number of molecules X, right? And this, there's still noise in this equation, right? So you can, so this is this is a launch event formulation where this is the this is the amount of X in a given time step evolves by we have our deterministic growth. We have an annotation for this. We have our deterministic growth and decay here, which is just the expectation value of how much you would get in the next increment. So this part of the equation people sometimes would refer to as, some people who come from physics like calling these things forces. Some people like calling this flow. This is the deterministic part of the equation. And then we also have noise, right? So, and in this example, this noise, takes this particular form that's imposed by this sort of birth and death process that we wrote at the beginning. Yeah.
So this, in effect, you're trying to basically parameterize these interactions.
You can say so, yes. So yes and no. So I'll show you. So at the end of the day, what these methods do is learn a neural network for G, right? So you do not get out at the end of the day a symbolic representation of G, although in principle you could if you fit G over a functional basis, for example, instead of putting, so G can be, you can, I'll say more about this in a slide or two, but you have flexibility about the choice of, for example, function space. So right now, we're making relatively few assumptions about the correct functional form. And the idea is to learn something useful from the data. And so, depending on what you ask, what you try to what you try to fit for G, it can give you different types of information afterward. Yeah.
What are the variables M and V?
Yes. So little m is just a rate constant, and V is the volume of of the cell.
The volume of the cell, is that what you said?
Yes.
Okay.
Yeah. So
Like
it's
the
right.
amount
So
of
it's just-
mRNA
it's-
or something.
Exactly.
Okay.
It's just it basically scales the amount of noise because the larger the volume, the closer it is to sort of fully deterministic. And small. Okay. So this is one example of a very general class of models, which are in the physics literature usually called diffusion models, and some of this other people sometimes say drift/diffusion because some people think of just this part as the diffusion term, and so the other one, the deterministic one, is the drift term. So what's going on here, right? So we have the independent variable is x, which is again, it's real valued in some number of dimensions. And so this in most of these single cell examples, this will correspond to gene expression across your D favorite genes, usually scaled in some way. So this is the state of the cell is characterized by its gene expression at a particular point in time. And then F is some nonlinear function that we are going to want to learn, which represents, again, these sort of deterministic forces or flow that tell you how different genes influence one another, right? So it takes in the entire set of gene expression values, and so any one gene can push up or down any other one gene at any sort of point in the phase space. And then W is a standard Wiener process, and that simply means that you can think of it as at each little time increment you choose a Gaussian random variable, and then G is a noise function, which again can have some correlation structure if you like, and so in general, right? Also, and this stuff comes up in various of these things. So G, in general, could depend on the state of the system also. Okay, so a brief mention. So this is a very, this isn't really a model, it's sort of a class of models. So it's a very flexible sort of, it's a very flexible model. And another way to think about it, so I motivated this as this is a natural choice from starting from a gene regulatory network. You can also, if you prefer, think of it as simply, we think that there's essentially some deterministic component and some noise, and this is one of the simplest ways of modeling this. This model is quite general, but it doesn't include every possible thing, right? So in particular, it's Markov, right? So the basic assumption here is that your current state determines what will happen to you next, up to this noise term, right? So that means that there's no memory, right? A second thing is that it's a first order equation, right? It's damped. There's no x double dots anywhere. There's no explicit dependence on your velocity, right? So so all of your where you go in the next time step is purely determined by where you are right now in the state space. And then another thing to mention is that, right, implicitly all of these models assume that little f here, this function, and as well as big G, are only allowed to depend on the variables you've measured, right? And so effectively that does mean that so almost certainly there is other variables that might be important that you have not measured, and to the extent that exists, it's sort of somehow going to get subsumed into this noise term. But the assumption is that you have sort of a good enough set of variables that you can get something meaningful from sort of making this assumption that essentially they are the things that influence one another in this deterministic F term. And as a brief mention, so the way that I've written this, F does just depend on these state variables. There's some of these methods for which that also allow for non-autonomous terms here, meaning like, for example, if you wanted to add a signaling factor at some point in time, that would come in as maybe an additional just constant term, but that you modulated over time somewhere in this little f. So some of these methods allow for doing that, some of them don't.
Yeah, one quick question. So you mentioned that this has this first order Markov property.
Yes.
The thing is that, say, if you want to step ahead in time using something like finite difference there, so then all the time steps will eventually contribute to a particular time step, right? So you time step from 0 to 1, 1 to 2. So if you step ahead in time in that manner, so the property at any point in time isn't really dependent only on the previous state, right? It's dependent on all the.
No, it's only dependent on where you
Probability
are.
of going where you go next. I'm sorry.
It's fine.
Okay, the probability matrix is defined only by the present state.
Correct. Yeah.
Okay, got it.
Sorry. No, that's fine. That's good. Right. Oh yeah. So, and I did want to mention partly because I'm actually interested in this stuff for applications that are not necessarily single cell RNA sequencing. That there are. This is very general, right? And there's other interesting systems that one might want to attempt to sort of again do sort of a data-driven learning of underlying landscape, if you will. Just one example is that so RNA sequencing, single cell RNA sequencing is the most ubiquitous kind of data that but people are starting to collect things like imaging data, more proteomics, other kinds of interesting data, even in the single cell context, for which I think these things will be applied much more as we go forward. And then there's wholly other types of scenarios that you might be interested in. I just put up ecology just to choose one that's fairly different, right? So in that case, you for example, you've got communities of microbes and they interact with one another, right? Like some of them produce things that others of them eat, or they kill each other, or so on and so forth. And they have they die and they're born and so forth. So that's the source of noise in that particular system, right? And so this is another type of system where there's a lot of quite interesting complex dynamics that people often can observe, and would like to understand that can potentially be modeled in a similar way. Okay, so right, so in our specific instance, we are again we're dealing with these scenarios where it's possible for us to take samples of, for example, your stem cells that are differentiating in your dish, but once you sequence those cells, they're gone, right? So we have potentially time resolved, probably not in like extremely finely time resolved, but we have some samples in time of cross sections of these populations. So in other contexts, for example, in soft condensed matter research, where you have colloids and other types of things, it's more common that you might have such a system and where you actually have trajectories. And in that case, so there's this sort of Langevin formulation is equivalent to so you can think about the instead, if you want, the evolution of the probability distribution of so because there's noise in the system, where even if you start at a definite state at a particular time, where you will be at the next time is probabilistic, right? And there's some transition probability that takes you from your density at position y at time s to position x at time t. And if you have trajectories, you can fit such a transition probability. But if you don't have trajectories, then really all you have access to is the marginal distributions of these, rather than these transition probabilities. So this is simply that, given that you have an initial state, right? What is the probability of a particular other state at at time t, right? So what is the probability over x, probability distribution over x at some subsequent time t? And this is just so this is a Fokker-Planck equation, and it's just you just get it from marginalizing this transition probability Kolmogorov equation over the initial conditions. So you have some distribution at time 0, and you just integrate over that. And so really all we know from the thing that we can like estimate from data is P of x and t for some, discrete times t. And what we would like to be able to do is fit f and d, basically. So this brings us to force or flow instruments, right? So as I just said, the idea is that you use data-driven estimates of P to infer F, and in some cases also D. And as mentioned before, the goals here are to predict the future time evolution of a system, right? And the idea is that, once you have such a model, you can start it at points that you haven't observed experimentally and ask where it's going to go, for example, predict things like the effects of perturbations, and then maybe learn things about the underlying biology from the structure of F. And some of the types of choices you have here are, what function space do you want to use for F and D? As mentioned, so by far the most common choice now is a neural network, which is then very flexible with some kind of regularization, which is one of the sort of technical choices that is now then can be a very flexible representation. What kind of loss function do you want to use? So how do you decide if how well your probability distributions that your model is generating match your empirical observations? And then, how do you actually do the optimization? As mentioned, there's been really quite a bit, I would say, of methods development of methods for basically this in the last little less than 10 years. And okay, how do these methods differ from one another? So some of the main differences are that in many of them, the F is constrained to be the gradient of a scalar potential function. So that is, it makes it it's a restriction. It makes it easier to infer in general, but it might be appropriate for some systems and not for others. Another is that some of these methods don't have noise explicitly at all. So some are purely deterministic. There are a variety of constraints on what types of forms the noise can take, right? So, one of the tricky things here is that, you can't infer an infinite number of things at once. You have to some extent limit the flexibility of what is allowed to be fit. And so there are some choices there. Another is whether or not the cells can themselves grow or die, which depending on the context, may be important for a particular experiment. Again, as mentioned previously, is the system supposed to be fully autonomous, or is there the possibility of having some kind of external signals and terms that correspond to that? And then there's of course a range of additional technical choices that people make, like exactly what activation function to put on neural network layers, what loss functions to use, how to regularize them, etc. Okay, so and actually, why don't I pause there before I talk just a tiny bit about questions. Yeah.
Is this force like the force flow plus noise generalize all the diffusion models, or specifically for this one?
Yes. So I guess right. So I mean, the model that the thing that I wrote, this is just a logarithmic equation that is could be anything. And so this is very general describes like many different types of dynamics in many different realms of biology, physics, etc. So yes, and people would, as mentioned, like in physics, people like calling these things forces, but that would be true of sort of any system that can be modeled this way. Okay, so yeah.
Is there a clear distinction when would a gradient based description be useful and not?
Right. Yeah. So I mean, the main thing is that if you if you're limited to being the gradient of a scalar potential, then you basically can't circulate, right? There's no curl in the field, right? So in one can also say that it's just it's a limitation on the sort of what types of functions f can be, right? I think that in many cases of cellular differentiation, maybe this is reasonable. It's probably not that the idea that this would be like purely descent on a potential landscape, maybe not true, but it's maybe very reasonable simplifying assumption. And I mean, I think that really you would just have to look empirically whether or not your predictions improved if you were able to include terms that didn't arise from a potential, right? There's a number of other types of dynamics for which it's clearly inappropriate. I personally am interested in immune. One of the things that I'm interested in is immune responses, which are potentially excitable, right? So they, you kick them, they go off, and then they come back, and those are very, very clearly not derived from a potential, right? And another type of dynamics that is like extremely obviously not derived from a potential is anything like a cell cycle or circadian oscillations, right? These things have limit cycles. You cannot construct such a thing with a potential, right? And so that's like the sort of qualitative. So, scenarios where, you think conceptually that maybe a ball rolling down a hill is sort of sufficient, then that might be okay. Other types of scenarios, clearly not. Most things probably fall somewhere in between. Does that answer your question?
Because all cells divide and they undergo cell cycle. So
yes. But I mean, for these types of analyses, sometimes cell cycle genes would be sort of explicitly removed beforehand. So the idea in many of the analyses that people are doing, they are, they're interested in sort of the genes that drive what is thought to be essentially a sort of unidirectional process.
So you filter out and coarse grain based on what you're looking at.
That's right. I think for practical purposes, yes. Yeah. But I mean, if you were studying cell cycle, then a potential is clearly inappropriate, right? Right. Okay. So how do you have any idea whether any of this stuff works? One of the nice things that people have done is to benchmark some of these methods on a really neat kind of experiment where there aren't too many of these types of experiments, and this is a very nice one where what happened here is that the group that did the experiment started with some these are HSCs at day 2, I guess. Please come in. They transformed them with a large, diverse set of DNA barcodes, such that which get inserted into the genome, such that cell and all of its progeny have the same barcode. So then, what you can do is so now, you took your cells at day 0 and you sequenced them, but now you can sample at day 4 and you can identify little groups that people call clones, which were descended from some of your initial day 0 cells, right? So in the design here is that, you take your day 0 population, you split it into 2 wells, and then you propagate these forwards. So now, what can be done computationally is to, train a model on this first well 1 data set and then test it on the well 2 data set to see how it performs. So what is the input, right? So the input is you have a gene expression state at day 0 in that top well here that corresponds to a particular barcode. And then in days 4 and 6, people will call what type of cell exists in the population of clones that descended from that barcode, right? And then so you train your model to try to take your gene, that initial gene expression vector, and give a proportion of cells of these different types that will start from that starting point. And then you can ask across the set of measured clones in the test set, how close were you to getting that prediction correct? So I'm just going to quickly show the results from, again, this recent paper where they tried several of these methods just to give you sort of a ballpark of what these things look like. So the first thing that they did was actually just to regress to try to make this prediction using linear regression. You might say, oh, well, why are we doing all this fancy stuff if linear regression works? In some sense, what the question that this is essentially trying to answer is, as mentioned, like, it's not obvious that the state of the cell at day 2, should be predictive of anything, right? I mean, if all of the cells were identical, which, maybe even at some earlier time point, maybe they even are, right? Before they've even maybe diverged from one another even a little bit, it might be impossible to maybe this gene expression state contains no information about the subsequent fates. So one way of looking at this sort of first thing is, it is still just a linear model. One could try something fancier, but basically this is addressing the question of, like, well, how much information does this initial gene expression state have in it about what the proportions of the final cell populations will be? And so the answer is, a lot, but not everything, for sure. And then the question is, how much of, now if we ask for this much fancier thing, which is an actual sort of a generative model of the sort of dynamics in the process, how much of this does it capture? And so this is the comparison of cross models, as you can imagine. So this last one, of course, is the one from this paper, which of course has to be higher than all the other ones because that's how these things work. But one thing that I was going to mention is that Sam is going to talk a bunch more about this. So there's a family of techniques that actually use an additional auxiliary piece of RNA sequencing information, which has to do with the ratio of immature to mature RNAs through information about whether they're spliced or not. This is called RNA velocity. These methods actually do really badly. This is just what these people found, and it's just, again, I think Sam will talk a lot more about this, but the RNA velocity stuff, a very cool idea, I think, and very sort of tricky in practice. Okay, that's all I wanted to say about the overview. So do people have questions about this before I talk a little bit more about sort of how one of these methods actually works? Yeah,
I guess maybe more about that, like, linear regression benchmarking.
Yeah.
I mean, like, you can do all the same, like, perturbation techniques that these methods do, also just in the linear regression case.
That's an interesting point. it would be, I agree with you that actually it would be, so I have not seen somebody try the perturbation experiment just using linear regression. And yeah, there's no reason you couldn't, right? Yeah, it would be interesting to see how that works. So this is specifically just like the cell fate prediction task. I kind of agree with you that if linear, okay, so I think to some extent I'll come back to this in a few slides from now. But I do think in an ideal world you get substantially more out of these models, but I agree with you that I think in many cases, if you can get both sort of prediction and the perturbation thing, then it's not obvious that all this fanciness gives you much more than your linear regression model.
So is linear regression the best baseline that you could have? Because the thing is that say when you do differential gene expression, you use a different underlying distribution, right? That corresponds to
Sure.
the transcript counts. Like
Yeah, this is what they chose. I think it's a perfectly reasonable choice. You know, you could certainly cook up a fancier model, right? I mean, my honest guess is that so you could fit a neural network, right? My guess is that this would do a little better, but not that much better. But there's no, I don't, but I haven't tried it, so yeah.
I'm not sure if you mentioned it since I was late. I want to ask that, what about the delay between the expression of genes? Means some genes have some delays to the other.
Yeah, so there are all sorts of, let me just flash back to the sort of mechanistic, I don't know if you were here when I did this. So this is a sort of mechanistic justification for writing down such a model, right? And it, as you say, right, it doesn't have, so, here the idea is that, you're just measuring the RNA levels, and the idea is that this is a good stand-in for example, the level of these proteins that maybe actually regulate. There are no time delays here. So there's a lot of stuff that's not included, right? So again, I think that in some sense, the scenario that in many of these cases, actually the data input is in some sense even further removed from such a mechanistic model, in the sense that oftentimes the variables people are actually working with are, for example, the projection onto the top 50 principal components of some kind of gene expression matrix, where typically first it's been filtered a bit for genes that have enough expression, are variable in the experiment, this kind of thing. But so it's I think that the connection between a sort of true mechanistic model and the variables that this model is dealing with are somewhat loose. And the hope is essentially it's still a useful exercise. But I don't think it's obvious that is the case, right? And the question about which are the most important things that are missing, I don't know that I have a great answer, but I think I think that at least in some cases this can be useful. And I think I'll show in a second one that I think is reasonably convincing, but I think also shows potentially some of the limitations. All right. Okay, so so this other questions. Okay. All right. So and I guess what are we going to? 1:30? Is that right? No, 1:45?
1:45.
Okay, yeah. So we've got tons of time. So feel free to ask questions. And we don't have to go all the way to 1:45. But yeah, good. We're not constrained. Yeah.
Sorry, this might be a silly question, but most of these models that you showed, maps one population to another.
Yep.
How accurate are they for, say, if I were to pick one cell and predict, what would be its, future state?
Well, the idea, okay, so the idea is that, right, so the idea is that you essentially train on this distributional data, and then the model is then able to make predictions where, you can give it any state. And in fact, there's, and in fact, like, so you might expect that, in, for example, regions of the phase space where it hasn't seen very much data, it would be very inaccurate. And in general, this is probably true. I can tell you that my group has played with some essentially sort of toy models that are quite simplified relative to these types of scenarios, but I have been actually a little bit surprised by the extent to which, at least in some cases, the sort of smoothness and regularization you actually sometimes can do some things in regions of phase space that you think you shouldn't be able to. But in general, like in principle, at least, if you're starting from a point that was reasonably well sampled, then yeah, you should be able to do this perfectly fine, right? There's no, it will, so your input is effectively sort of a delta function, but then it will predict the subsequent probability distributions, which will be some kind of distribution over time. Does that make sense? Okay, right. So again, so I'll just going to go through, not in sort of exhaustive detail, but hopefully enough to give you a flavor of what's going on. One of these methods a recent method that I like. So this is from a recent PNAS paper from Manu Shardez and Michael Shelley in New York. The goal here is as illustrated in this very simple example, which is actually only two-dimensional. So here there are variables x1 and x2, they cross-regulate one another. And at the end of the day, so you start with some points over here, and then at the end of the day, you can either end up with x1 high and x2 low, or x2 high and x1 low, and the thing evolves over time. We have our population that started at those black spots, and it's sampled at a couple of different time points here, just T2 and T3. And the goal now, the goal now is to fit F and D. What the main innovation of this particular method is basically to borrow from a bunch of, there's a bunch of breakthroughs and recent progress in diffusion modeling for general AI applications, which I'll talk a little bit about the relationship between this and that at the end. But there's some nice technical things that can essentially be ported over. What this method does is takes advantage of the fact that there's an equivalent way of writing such a Fokker-Planck equation called the probability flow ODE. And this is actually just a very simple rewriting of this equation at the top here. And what it does is, what this thing is, as you can see, it's an ODE, so it's a deterministic equation for these x's. And what it does is, the probability distribution over these x's is the same as the probability distribution over the stochastic process, but they flow smoothly. This diffusion term, the noise term here contributes an extra term, if you will, to the flow in X, which, in a case where you had, if you had simply, just this sort of diffusive process going on, it would make your points, it would generally make your points spread apart from one another. So it takes care of the fact that there's this extra there's this extra sort of spreading of the probability distribution from the noise over time, which turns out to be proportional to this gradient in X of the log of the probability distribution. So the main technical, the main reason that this is now useful is that there was a relatively recent technical breakthrough in which people figured out, so this is like a kind of a weird fancy object, this del X of log P of X thing, but people figured out how to estimate it to actually be able to get this from empirical data. So this is something called, well, so one of the ways of doing this, which is, to my knowledge, the one that's typically used in, Dolly and whatever these AI diffusion models, is called slice score matching. This main paper from 2019, which, I'm not going to go into the details, but basically there's just now a way of training a neural network to estimate that function. And so now the way that this approach works, so you have your points at a particular point in time, and you have your current model, which includes which is written right here. You have your current model, which has whatever you have as F right now, whatever you have as D right now. You've estimated your score function. And so you can simply update your points to the next time point. And then you want to measure how far off you are from the actual set of observed points at that time point. And the typical choice here is a metric based on optimal transport, which is something called the Wasserstein or Earth mover distance. And this is really just a way of comparing these two probability distributions. And then so in this method and in most cases, the loss function, the way that this thing is penalized is simply by adding over all of these distances for the whole time series. So in this case, you can imagine doing this in various ways. In this case, the way that it's done is simply that points are advanced from time k to time step k to time step k plus 1, and then the distribution at k plus 1 and the predicted and actual k plus 1 are compared. All right, so again, this is an example a little bit more explicitly of how this works in this toy example, right? So in this particular method, this neural network, first there's a neural network that's trained to estimate the score thing. That is fixed, and then the neural network F is learned, so importantly in this, and then so this whole module, right, acts as a function essentially, which allows you to simply compute the thing at the top and advance your points forward, and backprop different, do your optimization. Using all of the lovely modern computational machinery for this kind of thing. What was it? Right. So importantly, in this method, so the form of the authors choose essentially a model which gives them a specific form for this noise term D. Right. So the one that they will use on the data that I'll show in just a couple of slides is actually this, what they call this chemical Langevin model, which I showed a few slides ago. But this means that effectively you only need to it's sort of written as two functions D and F here, but really you're only inferring effectively sort of one nonlinear function, which in the example as I said in the example that I showed earlier, is this function little g, and that gives you both the deterministic and the noise part. And so that here they have a few different sort of principled models for what the noise might look like, and they compare them, how they work, both in this case in some instances in cases where they've simulated and they decided up front which was the correct noise model, and then eventually in data. Right. So this is an example of one of those exercises. So they used this chemical Langevin. They used a Gillespie algorithm to simulate exactly these chemical reactions, using, in this case, this network of 5 transcription factors, and then they compared how well the inference did on using the correct noise model, which is this CLE one is the correct noise model, and then using a few different other noise models. And so the point here, which I think is kind of interesting, is that the first plot is essentially is just a comparison of the inferred F. So the inferred, so I should say, you're right. So the output of this at the end of the day, you have a neural network, which is this potentially complicated nonlinear function, but you hand it a gene expression state, so it's a vector in RD, and it gives you out a velocity vector, right? So it tells you, so it does not give you, an equation for what is the flow at that point in the state space, but it always gives you an answer, right? It gives you a direction that things flow at any given point in the state space. Okay, so they compare, so here what they're doing is they're comparing, so they know ground truth what these forces or flow errors are supposed to be, and they're measuring a root mean squared error from the inferred F to the actual F, and so this is as a function of this cell volume parameter V, which controls how much noise there is in the system. So the main point that they make is, so the correct noise model does the best, right? But the other ones, sort of don't do too badly, one could say, right? I mean, they all learn something that gets better as the thing gets more deterministic. And they all learn to approximate these arrows reasonably well. But the point that they make is that actually, interestingly, they do this by, they actually get the interactions between the transcription factors somewhat wrong. So in this case, the way that they are measuring what an interaction is that they compute the Jacobian of the inferred F, and then they average it over the phase space. They actually use a slightly different measure of interactions later. I don't know, there's maybe some art to deciding exactly how to quantify the extent to which, something represses or promotes something else. But the main point that they make here is that basically, the correct noise model actually does recover the structure of this interaction matrix pretty well, whereas the incorrect ones are just actually completely wrong. And so sort of a similar message here. So this is yeah.
So they're just taking RNA-seq data.
Yep.
Right?
So this is not RNA sequencing data. This is a simulation of. This is simply a simulation. The next one is going to be.
Oh, I see. Okay, so this is a simulation with that model.
With that model.
I see. Okay.
They just simulated their model,
Okay,
so they
okay.
know.
Sorry,
the
I missed
noise
that
is
part. Okay.
this because they did. That's what they did.
Okay, got it.
Yeah.
Thank you.
Yeah. So right. So what? So okay. So you might say, and in fact, that how do I know which noise model to choose? I think that they're, so I don't know exactly how they would answer this question. I think one answer could actually be that to the extent that you can do internal checks, one can compare in instances where you aren't sure. They do seem to like this chemical Langevin formulation for gene expression. This is again going to be the one that they also do compare in this. They compare for this actual data set that they use. They compare it to their other noise models also, as well as to one of these other methods, which is called prescient. So prescient is prescient is infers a gradient of infers a potential effectively. So these things, unlike this method that I'm talking about now, the PFI thing, they are limited to a gradient of a scalar potential. The forces must be the gradient of a scalar potential. And then the other main difference is that the noise is limited to being additive. And so, right, so this is a result for, this is RNA sequencing data. So what they did was, as far as I can tell, actually it was not completely unambiguous to me from the paper exactly what they did, but I believe what they did is to take 14 known transcription factors for this system and essentially use the levels of those as the input. So if you look at the first two principal components in this already reduced dimensional space, you see this very nice splitting where this is actually, again, this is a part of HSC differentiation again. We're going to get megakaryocytes and something else that I don't remember, but and this is the next panel. So the experiments, the cells were sampled on days 0 through 11 here, so, some sort of nice time points. And so this is just an example of now resimulating from their fitted model and from the prescience model. And so, you can see that like qualitatively, obviously their one looks a lot better. Another couple of comparisons. So they look at the levels of these different transcription factors. This is the correlation between the predicted final state for the erythrocytes and the megakaryocytes for their method and prescient at the final time point of the experiment, which is day 11. And so so this makes sense to some extent from these plots. In general, the correlations for both are pretty decent. With their method performing a little bit better. But the real difference is actually in that, so in this particular instance, there is what they say is essentially a known set of regulatory interactions, which is drawn in this little cartoon here. And so they think they have some biological knowledge of like what actually are the interactions between the transcription factors. And so they try to measure, they measure measure these interactions in their system by taking one of their genes and turning it off and then measuring its effect on the other genes at, I can't remember if it's all time points or the final time point, and then measuring a cosine similarity between that output and essentially a simulation of this. So the sort of main message of that is that what form of the noise is put in here actually really matters for whether the sort of correct interactions in the system are recovered. So that basically like there is a number of different versions of F, this sort of deterministic interaction matrix, that will reconstruct for you, that will do a decent job of showing where cells go and maybe making things like fate predictions, but the structure can be incorrect if what you want to do is sort of learn additional things about the biology. So here what happened in their thing is that the one of this chemical Langevin formulation of the noise actually does pretty well in terms of actually recovering a picture that looks like relatively like the known interactions. Probably not that surprisingly, this sort of square root of X, which is somewhat different, but has sort of more quantitative overlap, I would say, with this red one, does it kind of okay. The additive noise model basically fails, and the deterministic and prescient really fail quite, are just inferring something that's entirely different as far as the interactions go. Okay, so that was all I wanted to say about that. Any questions before?
If the data you have is sparse, wouldn't the score function be undefined, I guess?
Yeah, so right. So yes. So this is an interesting question that actually my group has been thinking about a fair amount, because we're interested in doing things that are a little bit like this, but where each of our data points is actually an entire zebrafish that we've sequenced rather than a cell. And so we have access to a large number of these little tiny fish larvae, but not as many as you would for cells. So we actually have applications where, you're not necessarily going to have tens of thousands of points. So my answer to this is you can do pretty well with a Gaussian approximation to the score. There's actually even a paper called The Unreasonable Effectiveness of Gaussian's Score Approximation, or something, some title that's similar to that. So there's, I mean, this is like a technical thing essentially, but I think there's quite a big difference between including the effects of noise, that is the fact that this probability distribution, there is this sort of spreading effect essentially. So including versus not including that at all, and so you need at least some information about what your distribution looks like at the previous time point. I actually think that you can get a fair distance, even if you don't have an incredibly well sampled distribution. But yeah, that's my opinion about this question. Yes, I mean, in principle, so I mean, I think at some level, so the slice score matching, the way that it works is you essentially try to estimate the distribution by simply taking a finite number of cuts through it along lines. People, I think, were surprised by how well this worked initially. But again, the thing is that given there's a question about, how badly do you do if you, right, you're trying to estimate the gradient in X of the log of this probability distribution. If you get your probability distribution off by, in some ways, right, how badly does that screw you up? Well, as long as you have some information about the width and so forth, maybe not too badly, right? But yeah, I think it's a good question. Okay, so right, so a few follow-up questions that might occur to you. Okay, so a number of these methods have optimal transport in the name, and this is what is optimal transport? So optimal transport is a set of is essentially the mathematical problem of how you transform one PDF into another while minimizing a specific cost function. So there's a lot of mathematical development of this in the 2010s that sort of allowed it to be computationally tractable to compute something called the Wasserstein or Earth Remover distance, which essentially so you want to now compute if you if you now want every piece of mass to be able to move to some other piece of mass in your new probability distribution, such that you minimize the sort of overall sum over all of those moves. That's what the idea, the Wasserstein distance is so that, and then that's your distance between your two probability distributions. So there's a number of and so all of these flow inference methods need a way to compare probability distributions, and so this is a choice that makes sense. Personally, I don't think it's super essential to, I mean, there are other ways of comparing probability distributions. Again, this one probably makes sense for this problem. Is it really so much better than using KL divergence or something else? I don't have a super strong or informed opinion about this, but that's sort of a summary of that. Okay, and then another thing that comes up a lot in single cell sequencing papers, and I think is a really cool idea, but has a lot of trickiness with it, which I think Sam is going to go into quite a bit in the next talk. It's an interesting topic. So one of the kind of interesting things about these single cell sequencing measurements is that there is a little bit of, even though it's a snapshot measurement, there's a little bit of temporal information in the raw data. And that's because any one of these experiments will typically capture some RNAs that are not yet mature. And you can tell that they're not yet mature because they're not spliced. So the experiments weren't designed to capture these things to begin with. And so, is partly why this is actually, one of the issues here is it's a very noisy, it's even noisier than gene expression values. And potentially also even more like variable from prep to prep and things like this, because again, these things, the experimental techniques for the most part, like essentially got all these transcripts by accident. However, it's very tantalizing because in principle, it could allow you to infer get an empirical estimate for these velocity vectors effectively, just from a single snapshot measurement. And there's many good, there are many instances, right? So I've been talking mostly about experiments that occur where you have cells in a dish that can be sampled from at different time points. There are many, many different applications that people are interested in where you simply cannot access an actual time course, right? And so it would be neat to be able to use this information. As mentioned, it's just tricky, and at least as shown here, I think that people who have played with this a bit, one should be careful, especially with the sort of off the shelf techniques, because whenever it seems like whenever you check them in an instance where you can check them, there can be issues. So I think Sam will talk a bunch more about that. And what one might also do. Yeah.
One quick question about that comparison between OT and KL divergence.
Yeah.
So as per as based on mine, so OT also gives you the path from one distribution to another, right? While KL divergence is mostly is
just a number. Yeah. Yes, that's true. And in some of these methods that actually, so the Wasserstein, excuse me, I was gonna say the Waddington OT paper, there's a couple of these methods that actually do explicitly use this. And I would say that, there's a Cell paper from 2019, which is the method is called Waddington OT, that is more grounded, like explicitly in optimal transport and using specifically these optimal transport maps from time point to time point. Most of these methods just use it as a distance metric.
Yeah, if you see like models like cell OT and cell flow, like
yeah, so right. So I think I'm not sure I'm familiar with those specific ones, but yes, there are some of these methods that do use more of this. This is okay. Okay, and so the last thing that the best thing I wanted to say is which I think is kind of neat, is to talk a little bit about the relationship between this and the diffusion models that people are using, all over these different AI things, for example, to generate images. Okay, so nowadays, in this context, what do people mean by diffusion modeling? So the idea is, you are Dolly, let's say, and you want to make a picture of a dog. but you know, you don't want to make a picture of a dog that's just like literally a picture of the dog that you pulled from the internet that already exists. You want to make a picture of a new dog, but it's supposed to be a dog, right? And so one way of stating this problem is you want to sample from the dog distribution, right? So, you know, where now your high dimensional vector is simply a vector of pixel values across your image, and you want to generate something that looks like a dog. And the idea is, right, that there is some underlying dog distribution, which is, it's complicated and high dimensional, but maybe you can learn it, right? And so the way that people have figured out to generate samples from such a distribution is essentially this type of diffusion modeling. So what's done is you take many pictures of dogs and cats, or sorry, let's just start with dogs. You take many pictures of dogs. You generate training data by progressively injecting noise into these pictures, right? So you generate things that look like trajectories, right? So if you collapse you have a long vector that corresponds to each of your pixels of your dog picture, and you add at each time step, you add a little Gaussian random variable. This defines a trajectory in your state space going out, and at the end of the day, you have something that looks like noise, right? But it's not exactly noise because it started from somewhere. And now what you can do is to do similar type of computational problem that we've just been talking about, to take now your training data, right, consists, for example, of the series of images that you produced, and you can train a model to estimate this deterministic force or flow field F. And so in this case, the computational problem is actually a bit simpler because you purposefully chose a form of the noise that you know completely, and it's a very simple one, right? But so now you infer F, and then you can run the process backwards, because you can use the Kolmogorov backwards equation. So now you can take a random seed that is purely Gaussian random noise, and you run the dog F. And it gives you something, and it doesn't give you, it's never going to, because you corrupted the image, right? It's never going to give you back exactly the same dog. But that's not what you wanted, right? You wanted a slightly different dog. And it is a very good way, of now generating something that at least, that looks like a new dog and, is potentially like a good sample from, this dog distribution. So this, in some sense, this is actually quite analogous to the problem that we were just talking about, right? So you can think of the noisy image, right, is your stem cell. There's small differences in terms of where you start that will eventually determine where you end up. And again, from a technical standpoint, in some sense, the problem in many of these biology contexts is a little bit. It's harder for a couple of reasons. One of which is that you don't have access to like infinite training data that you can just generate very easily on a computer, and another is that your noise could be substantially more complicated than the simple types of things that you would use in this other context. But the fact that the remainder of sort of that process of inferring F and so forth is essentially the same means that methods development in this big world of computational work on AI can actually port over and be useful for some of these biology problems, and that's what this probability flow inference paper does. And so then a final thing that I'll mention is that a large difference in this context is that for this image generation problem, at the end of the day, you don't care what F is at all, right? It's just a way of getting you the image that you want. So there's no sense in which there's a sort of correct F, and if it works, it works, right? Whereas one of the reasons that I think one of the motivations for doing this kind of landscape parameterization in biological systems is that we are actually interested in what are the underlying biological interactions, and can we learn something about this from this function? So finally, I just wanted to throw up. So you'll Eric Sigias will be here in the next few days, so you'll hear some about this. But I just wanted to mention a couple of other threads of that might be interesting. So the first is maybe that you think this idea of inferring low-dimensional landscapes that might underlie cellular differentiation is interesting, but you don't totally buy all of this fancy modeling, which I think is a very valid perspective. And so I think this group has done really a lot of really interesting work with some similarities, but a number of differences from the stuff that I've just been describing. And then finally, I have certainly not read this paper in detail because it just came out last week, but it looked quite nice. So a reference to a paper about inferring dynamical systems types models in biology that talks more about some things like Koopman operators and so forth that are some of inferring functions over other bases and things that are primarily a little bit more classical. It gets to some of these more AI-driven things at the end. Again, I have not read this in detail, but it looked like something that would be potentially useful as a counterpoint. All right, so just to summarize, what we talked about is that is this family of methods that people have developed recently for diffusion modeling in contexts where we have destructive time series measurements, and we're interested in doing things like predicting fates of cells and being able to ask what the effects of perturbations might be, and then finally also potentially to ask something about underlying regulation that's driving these types of decisions. This is one can think of this as one example of trying to do direct inference of an underlying dynamical model from high dimensional measurements. One of the reasons I think this is interesting is it does really leverage a number of recent advances in applied math and computer science, and again, so these inferred functions are in most of these cases they are sort of these neural network black boxes. That's how these methods work. It isn't like a necessary. You could infer other types of functions, or potentially do some kind of sparse regression on the space on simulations or something like this after you've inferred something like a neural network model if you wanted a symbolic model. They can nonetheless encode sort of things about for example the interactions between different transcription factors.
So how do you, so is there a way to quantify the effect of an intervention in that process? Like,
yeah, good
can
question.
you encode a drug or a gene knockout?
Yes. So okay. So the way that gene knockouts are typically, people handle gene knockouts like a couple of different ways, but the simplest way to think about it is you simply set the expression of that gene to 0 at a particular time or for all times, right? Which is just something you can do by hand. And so that's sort of easy in the model. The effect of something like a drug intervention is more complicated. I think that what people, if you're interested in this or like a growth factor or something, right? I think what people would typically do is to allow the F to have non-autonomous terms. So to have like explicitly, for example, a term that's fit that for the effects of that factor, and then try, for example, to you have some experiments where, for example, it's introduced for some time window at some concentration, right? And you try to learn, but then you might try to learn like the effects of this in general from this set of observations that you have, right? And so you might try to, so you would potentially try to infer like such a sort of source term or some, so like this drug might, for example, promote expression, like make a particular set of genes grow, right? And so you might, so you might try to infer like, and you would probably have to choose sort of a fairly simple functional form for it, but like, a simple thing is, that it would just like, this drug makes these genes go up over, with maybe some saturation, right? Something like this. So I think, so you would, that's the way in which I think one would try to incorporate such a thing, is, to essentially explicitly include extra terms to correspond to this input in the little f function.
Yeah, one
Does that
more
make sense?
follow-up question. Yeah, there's one follow-up question. Like, so how generalizable are these models? Let's say, if is there a way for us to have a pre-trained model that someone can use in their
that's a very interesting question.
in a new cellular context or like perturbation contexts?
This is a very interesting question. I don't think my guess is that I think one can ask whether or not they even totally work on in the context in which they were trained. I think my guess is that they don't really work except in the specific on the specific this particular kind of cells. I think you could certainly hope that it would work on my cells in my lab and somebody else's cells that are supposed to be the same cell type in their lab.
Yeah, I can understand that.
But I think that I think so people dream, in people's wildest dreams, this like it is possible to train such general models that would predict that you could do this. My personal opinion is I'll believe it when I see it.
Thank you.
Yeah.
First of all, thanks for the talk. It was really good. This is again another technical detail, but for this type of problem, what was the initial motivation to try to use something like score-based?
So this
what advantage does it provide?
Right. So the main thing that so basically like it's just it enables you So it makes it easier to this deterministic ODE like just makes the inference problem easier, and so it means that it's easier to you have more flexibility, for example, for what you can have D be, and the inference problem is still tractable. That's I think that's makes the whole probably also makes the whole computation faster, although I'm not sure about that. But it's it just makes the practical aspects of like actually doing can you, for practical purposes, perform the optimization? Can you get it to converge? Does it work, right? It makes it so that that's true for larger classes of choices for D. Yes.
Just to make clear for myself, I know that RNA sequencing, that distribution does not obey Gaussian. They are
absolutely
negative binomial.
Yes, typically negative binomial. Yeah. So you're right, absolutely. So so absolutely. But this is part of the motivation for choosing not choosing an additive noise model for D, right?
Mm-hmm. But in second slides, we were talking about modeling noise of RNA sequencing by Gaussian probability distribution. I want to know Gaussian probability distribution where? Maybe I'm making mistake. The part that you were talking about if the samples were rare.
Okay, yes. All right, sorry. So that's all right. So this is me editorializing on the side. There's these methods, I should be clear. Okay, so so this is a good point. So you know, so that was a comment that came from actually. So these methods, these methods, at least this PFI method, you know, very much does not assume a Gaussian distribution, right? And there's all this score matching built in such that you can infer a non-Gaussian distribution. Those comments had to do with a different context of modeling that, you know, that where this is like maybe a more appropriate choice, but isn't here. This stuff, none of this stuff assumes a Gaussian distribution, and whether or not that is appropriate, would depend on context.
I think that might be somewhat tied to the question I had regarding use linear regression, because the underlying assumption is that you have normal, but
that
again,
the noise.
negative
Yeah, I mean,
binomial
so
is
you know, it's like so I mean, concretely, right? Like a lot of people will typically take a logarithm right before they of their RNA sequencing counts to make things more Gaussian before doing right. So you know, it's of level, you know, it's maybe not so bad in some contexts, right? But again, one can discuss whether or not like this is truly appropriate and whether it's really screwing things up and so forth. Yeah. Okay, thank you. Thank you.